Onchocerca volvulus is a nematode that causes onchocerciasis and is the second leading cause of blindness worldwide after trachoma. Onchocerca volvulus is primarily found in sub-Saharan Africa, and humans are the only known definitive host. It is spread from person to person via female biting black flies of the genus Simuli. Onchocerca volvulus parasites obtain nutrients from the human host by ingesting blood or by diffusion through their cuticle. They may be able to trigger blood vessel formation because dense vascular networks are often found surrounding the worms. Humans acquire onchocerciasis through the bite of Simulium black flies. It is because the fly develops and breaks in flowing water. Onchocerciasis is commonly found along rivers and is sometimes referred to as river blindness. Onchocerciasis causes severe itching. Long-term infection can cause keratitis and inflammation of the cornea in the eye and ultimately leads to blindness.
First, the skin snip biopsy. The biopsy is performed using a sclerocorneal biopsy punch or by elevating a small cone of skin with a needle and shaving it off with a scalpel. This will result in the removal of around 2 mm of the tissue. The tissue is then incubated in normal saline at room temperature for 24 hours to allow the microfilaria to emerge. The microfilaria then can be identified under the microscope. Next, if a patient has skin nodule caused by onchocercal infection, nodulectomy can be performed for the identification of the microfilaria in the tissue. Slit lamp eye examination also can be used to visualize microfilaria in individuals with eye disease. Lastly, serological tests. They are antibody tests that can assist in the diagnosis of onchocerciasis. This is a general screen for any filaria infection. Because the test is highly sensitive, it is useful in determining if an individual has had filaria infection, but it is not specific enough to identify which filaria infection. With the antibody test, the results indicate only that the patient has been exposed to the disease, but it cannot determine if the patient has had active infection.